This video shows methodology for testing insecticide susceptibility using insecticide-impregnated papers on malaria vectors. Surveillance of insecticide susceptibility is very important because, along with insecticide efficacy tests in the field, it allows regional and national disease control programs to make decisions about pesticide use. There are two methods for conducting susceptibility tests: the WHO method, which uses tubes and insecticide-impregnated papers, and the CDC method, which uses glass bottles and insecticide. To learn more about the CDC method, visit their website. When the effectiveness of an applied insecticide in killing the mosquito vector is less than desired in the field, it may be due to operational factors, such as inadequate spraying or old mosquito nets or the loss of susceptibility in the vector population. Therefore, susceptibility tests are complementary to tests for the efficacy and residual insecticide effect. There are some general considerations to be aware of before starting the tests. These include: the places or sites of entomological surveillance priority should be established by the country programs, taking into account previous use of vector control interventions and available resources. WHO tests should be done at least every year in entomological surveillance priority sites in disease vector species for different groups of insecticides and those that are in use, including those incorporated into the fiber of the long-lasting insecticidal nets. There are two ways to generate mosquitoes for susceptibility testing. One would be to collect larvae from the field from the site where you want to test susceptibility, and to then grow them in an insectary and use the females. The other method is to catch blood-fed mosquitoes and to let them lay eggs and then raise the eggs to adult. Usually non-blood-fed female mosquitoes between 2 to 5 days old are used for the test. The temperature at which insecticide exposure occurs, ideally 27 degrees plus or minus 2 degrees centigrade, and relative humidity greater than 60%. WHO bioassays allow you to determine if a population of mosquitoes is susceptible to insecticides used for public health, which is evidenced by the death of mosquitoes in the population that are exposed to the diagnostic dose of insecticide. In this methodology, the exposure times and the intervals in which mortality are recorded are fixed. The interpretation of the results includes a range of mortality for use by decision makers related to vector control, which will be talked about later. Materials necessary for the WHO bioassay include the following: 4 exposure tubes and 1 control tube. Plastic tubes have screw caps on each side, along with a mesh covering on one end and a sliding plastic plate on the other, with a 20mm hole through which mosquitoes are introduced. The tubes with red dots are used to hold the insecticide-impregnated papers. The green tubes are for the papers without insecticide, control papers, and resting period papers. Insecticide-impregnated papers are used to line the exposure tubes. White papers with no insecticide is used to line the resting tubes. The papers are fastened inside the tubes with metal clips. The copper clips are used for the impregnated papers, and the steel clips are for the resting tubes. When the test is finished, the clips should be stored separately. Wear gloves to protect your hands from the insecticide, and make sure to wear a lab coat. If you need to go into the insectary to get mosquitoes for the test, make sure to wear a different set of gloves so that you do not introduce insecticides into the insectary. The control tubes are lined with paper that has been impregnated with the same solvent as the insecticide-impregnated papers. Before starting the bioassay, label the resting tubes and the control tubes. Then put the papers in the tubes, starting with the insecticide-free papers in the resting tubes, then the control, and lastly the insecticide-impregnated papers. Remember to insert the paper with the name of the insecticide facing out. Every exposure tube corresponds to a resting tube. Remember to use the correct tubes for the impregnated papers, those with the red dot, and for the control, those with the green dot. Using an aspirator, transfer 20 to 25 mosquitoes to the resting tubes. Then transfer the mosquitoes from the resting tubes to the exposure tubes. Exposure time is 1 hour for all insecticides, with the exception of phenitrathion, which is 2 hours. For pyrethroids, count mosquito mortality, or the number of mosquitoes knocked down, every 15 minutes during the hour of exposure time. Once the exposure time is finished, transfer the mosquitoes back to the resting tube for 24 hours. Separate the exposure tube from the resting tube. Attach a piece of cotton wool soaked in a 10% sugar solution to each resting tube. Under field conditions, the tubes with the mosquitoes should be covered with moist towels and kept for 24 hours in insulated coolers. Make sure to use different coolers to store the resting tubes for the controls and the resting tubes for the exposed mosquitoes. After 24 hours, count the number of mosquitoes dead or alive, recording the data on the information sheet. The mortality rates are calculated as follows. Control mortality, C, equals the number of dead mosquitoes divided by the total number of mosquitoes applied to the control tube multiplied by 100. Exposure mortality, E, equals the number of dead mosquitoes divided by the total number of mosquitoes applied to the exposure tubes multiplied by 100. If control mortality is between 5% and 20%, the exposure mortality should be corrected by using Abbott's formula, where corrected exposure mortality equals E minus C over 100 minus C multiplied by 100. If control mortality is greater than 20%, the experiment should be discarded. If the mortality rate is between 99 and 100%, the mosquitoes are susceptible to the insecticide. If the average mortality is 98% or less, it indicates possible resistance. To be sure of the results, it is necessary to conduct additional tests to verify that the mortality rate is consistently below 98. If the mortality results are less than 98%, then the assumption of possible resistance has been confirmed. Once this is done, it is necessary to use biochemical and molecular testing to confirm the mechanisms of resistance in order to establish a resistance management strategy. Biochemical and molecular testing can be done at a reference laboratory. As a precaution, the WHO recommends that a resistance management strategy is included in any insecticide-based vector control from the beginning. Once the test is finished, the mosquitoes should be stored in properly labeled vials. The vials should have a small hole in the lid to allow the escape of moisture. They are then packed in airtight bags with silica gel to keep them for morphological identification and for molecular identification if required. The information should be sent to the Ministry of Health and the National Malaria Control Program to support decisions in insecticide and vector control. You have now seen a general overview of how to perform the WHO tube test to determine insecticide susceptibility within mosquito populations. As with any methodology, practice is needed to improve technique. For more information on the standardized WHO protocol, integrated vector management, or training materials, please visit our website.